All right, the next series of mini lectures are going to be about chapters 2, 3, and 4. Chapter 2 introduces some biological chemistry. Chapter 3 is about water, a good molecule to start with. And Chapter 4 is about some other biological molecules. You'll see that biological chemistry is awesome, and because we get to focus just on a limited number of atoms and a limited number of chemical behaviors in life. Life is really rich and diverse, but a lot of its diversity comes from stacking the same Lego blocks in a variety of different ways, with the Lego blocks being little atoms and molecules. And so I wanted to start out with this image because we're going to be down at this level, and you can see the scale here. And I've got another scale image. I also put it on our handouts that I think is a nice one to conceptualize this. The atoms that we're going to start out with are invisible. They're smaller than the strongest microscope can view, and even most of the molecules that we work with are invisible under any kind of microscope. And it's going to be important to use some images from the book that we can understand how these atoms get put together from into molecules and how they work. But again, huge amounts of biology that many students are interested in, to study and work on for their whole career or to live in and enjoy happens up here, and that's more BIO107, and we're going to be down here at cells and molecules. And where we're heading is we're going to be going through the book. We have these 3 chapters in chemistry, and then chapters 5, 6, and 7 also fit together. 5, 6, and 7 are using molecules to build up to make a cell. And so chapter 6 is about cell biology. And so I think we have our 7 themes. And this is where we're at in our 7 themes. Is for our course, we're going to build chemistry to understand this, that all cells on Earth exist in a water environment. Doesn't matter if they live in the desert or not, they're still the immediate surroundings, and interior of a cell is always a water environment. And water has some special chemistry that's very conducive to life. We're going to learn about it. And then in that water environment, the cells, each cell is surrounded by a membrane. Some cells have more than a membrane, so they might have a surrounding cell wall and so forth, like in a plant. And lots of bacteria have very different types of cell walls. And animals have material called extracellular matrix that help support the cell in its environment. But the bottom line is we're going to learn that every cell has a membrane, a cell membrane. And then within that cell, there is similar chemistry, and a big focus is on proteins. And you hear about proteins in your diet, like we learned about in the first video of our course. We eat proteins, we digest them into amino acids, but then we build the proteins we want. And your body has thousands of different proteins, each with a different structure, therefore a different function, and they're put together by different patterns of the same amino acid building blocks. A protein has thousands of atoms in it, so it's called a macromolecule, just a big molecule. And we'll really learn a lot about proteins in this course. Our course is really kind of revolves around proteins. And so we're heading to some protein chemistry in the context of a water environment. And you see, we're just going to chunk into our course a big part of our goals. And this is about cells. So we're going to build a cell. And here's a picture of a cell from your book. And this is a cell that's called a eukaryotic cell. And eukaryotic cells are plants. All plants and animals like us have eukaryotic cells. And also there are lots of single-celled eukaryotes, like the yeast that causes bread to rise, for example. And so there are lots and lots of eukaryotes. Prokaryotes are examples are bacteria and blue-green algae, but when we think of prokaryotes, we think of bacteria. When we think of eukaryotes, we like to think about us. And eukaryotic cells are big and bigger, and they've got a scale here, and we're going to talk about that scale. There's some features in Chapter 6 when we talk about the cell that we'll get to. And within this, localized within this in various locations doing various jobs are going to be lots of proteins. they're going to be the tools of life, like that. Okay, so here's a handout I'll talk a lot about. So I posted on our website. If you look at our course website, well, let's just do that one more time as we're getting started with our course. See if we can get to it. So, you should bookmark this page because we're going to go to it all the time. Basically, you find Dave Champlin on the web or the biology department at USM, and then there's our course, and we've got these handouts. And so there's our themes. Then there's this image I'm just showing the sizes of stuff in biology, and in the next video we'll talk about some chemistry, and we've got a handout for that as well. Okay, so like I just said, there's a handout, and here's the idea. There's Boston. I call this the Boston metropolitan area. Okay, imagine an area that's 30 miles across. Okay, and then imagine that's one eukaryotic cell, one of your typical cells. And eukaryotic cells are often about 30 microns or 30 micrometers. Micron and micrometer are synonymous, or 30 millionths of a meter. And so here's from a different book, but I really like this scale. So people are often a couple of meters high, 6 feet high, like that, and tall, and then we scale down. Each of these are a tenfold. I guess that one's covered up, but 1 millimeter and 100 micrometers, and we're going down tenfold in each case. And so see, there's a range of sizes of plant and animal cells, and the cells in our body have various sizes depending on their cell type, and but they're about that big. And I tell students that here's a millimeter, and a millimeter is about the gap that you produce if you just try to pinch your fingers together and keep them separate. That's about a millimeter. And so we're saying that in a millimeter there would be about 30 cells. So imagine making a gap, right? And then making 30 little cells across it. Yeah, too small to see, right? You couldn't see that cell. Definitely, significantly smaller than the period at the end of the sentence. But still, you can imagine that size, right? That's a cell in your body. And, the cell is relatively huge. There's a lot of stuff going on it. So I say imagine blowing that up to the size of this Boston metropolitan area. And we'll talk about it. So, if a cell was actually 30 miles across, we've got this scale I put in over here. So we look down here. Inside the cell, we're going to see there are going to be some organelles. There are going to be some localized factories, and they're going to be about the equivalent of about a mile across. So like not as big as a suburb, but like as big as a neighborhood, right? A neighborhood in the cell. And then, if we saw a bacteria would be as big as a school. Right? So, imagine there's the Boston metropolitan area, and there's a school. That's about how big bacteria are, right? So eukaryotic cells are a lot bigger. And then we get down here, and these will be some pretty significant sizes here. So we got proteins, and there's a range of size of proteins, but proteins are typically going to be about this big. They're going to be about the size of a big truck. And that's kind of a nice thing to think about, because, there are a variety of different kinds of trucks, and there are lots of them in this metropolitan area. It turns out for each type of protein that we talk about, there are thousands of them. So, for example, you've probably heard of hemoglobin in red blood cells. There are tens or hundreds of thousands of hemoglobin in each red blood cell. So imagine that. This cell is crowded with proteins, a lot of different kinds of proteins, and lots of copies of the same protein. In our book, whenever it shows a protein, it shows it as a cartoon because it's invisibly small, and it just shows one. And students get a misunderstanding of the fact that there are actually lots and lots in every cell of a specific protein. And one thing to think about that's a kind of a weak thing about this is that this, imagine if it were a cell, would actually be more of a sphere. So we're filling up a sphere, not just trucks driving around on a flat surface, but filling up this space. So when I say there are 100,000 of this type of protein. You're like, whoa, that's going to fill up fast. If it's traffic, it's going to be really crowded. But it's 3 dimensions, so we have a lot to fill up. So that's a little bit of weakness, but still, I think it's a pretty good scale because it gives you a feeling for this. Imagine trucks driving around. And then we're going to get down to these atom level, and the smallest molecule we'll spend time talking about is water. And water is kind of nice in this analogy. Water is about the size of a watermelon. And this whole cell is really full of water. There's water outside too. It's mostly water. So imagine this space being mostly water and then crowded with all kinds of larger molecules floating around, diffusing around, moving around randomly inside the cell and outside of the cell. So we'll talk about this scale a lot, and it's important. It's important to get a feeling for scale. And on the test, I can ask you to rank these sizes. Which is bigger, bacteria or a eukaryotic cell? And we can have a series of comparisons on the test to have a feeling for the different sizes. Again, all this stuff is really little, but it's real, it's physical, and we can imagine it and get a very good handle on the physical nature of the cell and the physical nature of the molecules. So see, a really big protein we're going to talk about is a macromolecule, It's hugely complicated, and it's about the size of a truck. Like that. So it's cool. I think this is helpful. So we'll talk about it several times. Now, what the book does in chapter 2 is it just dives into it and says, we are going to take a look at the cell and we're just going to burn it down to the lowest, smallest scale to start with, and then we're going to build up from there. And that's this reductionism that I talked about, right? The whole is not greater than the sum of its parts for people that work in this field. They're like, yeah, no, we're going to tear it down, take this car apart, and look at it, every little piece, and understand the whole car. And that's the attitude, and so we just zoom down to this level to look at atoms. Everything is constructed of atoms. All matter is constructed of atoms, and certainly all life is constructed of atoms. Atoms, I already mentioned, are like Lego blocks. They have certain physical characteristics, chemical characteristics, and there are only about there are only about 5 atoms that are really prominent in biology. We'll talk about that, and more like 50 different atom types. I should say 5 different elements or types of atoms, and we'll get to that. And then more like iron and so forth that are pretty rare, relatively rare. We have lots of certain elements. So we'll get into that. Okay, so here we go with it, chapter 2. And for these chapters, these mini lectures they're talking right out of the book. I'm just re-describing right out of the book. In the first chapter, it was an overview, and I had some stuff I wanted to say. It was different, or different emphasis than your authors. But here, I'm trying to tell the same story, but it's worth it because it's a good story to learn well. All right, so we start out with an atom, and this can be any atom. Just any atom in biology, but any atom anywhere, in dirt and air, whatever. And so it has these 3 component parts, and physicists look at even smaller component parts of an atom, subatomic particles, but it turns out those kind of never come up in biology. They're significant, but biologists get everything they need out of studying these fundamental components of any atom. And so here's how it goes. We've got 3 different things that we got to learn about, and it's really straightforward. The first one are protons, and protons are located in the nucleus of the atom, and each proton carries a plus charge, and we'll talk about them briefly. Then neutrons have no charge, also in the nucleus. Then electrons orbit that nucleus and have a negative charge. Different atoms have different numbers of protons, neutrons, and electrons, and that's what makes them different. And so let's just look at this. Well, we'll get back to that. Okay, so let's just look at this. This is the periodic table, and it's just a part of it. Because biologists really deal with only a subset of the periodic table. And that's nice. In chemistry, you have to learn the whole periodic table, and there are a lot of exceptions, a lot of strange atoms out there, elements out there. Each of these boxes is a square around an element, and here are in blue, I highlighted are some of the most important ones. And there are really actually 5 that are the far and away the most abundant elements in life, in all life. So if you look at a bacteria, any bacteria, it has mostly these, lots and lots of these over and over again. A protein is made up of 10,000 atoms maybe, and well, thousands of atoms, and probably 98% of them are those 5, are one of those 5, and that's shown back here. This is from our book, and it just shows, yeah, here's some percentages. Just shows that these 5 make up lots, and you'll see these are like Lego blocks with certain building features that are just used over and over in combination and patterns. You've heard of some of these from our diets, right? They're essential minerals that we have to get in our diet, but we just need a little tiny bit of them, but we need them like that. There's some really great stories of how essential those are in our diets. So we'll focus in on this. Here's the first thing to say about this. The atomic number is the number of protons in the nucleus. And take a look how the periodic table is organized. There's the atomic number. It's the subscript to the left. And see what it's saying? It's saying that for each element, it has a different atomic number, a different number of protons. And that's what makes elements different. If the number of protons were to change, the element would change. That's something like turning lead into gold. Pretty hard to do, rarely happens, never happens in biology. Okay? And the elements have names: helium, hydrogen, helium, lithium, so forth, like that. So they're named, but it could have been they could have just been numbered. And it's really interesting to say lead and gold, because lead and gold are different just because they have different number of protons. These elements all have very different chemical characteristics, chemical behaviors, and it's because they have different numbers of protons, like that. This number is almost never written next to the atomic symbol because they're redundant. Helium always has 2 protons, so it's kind of nice to think about that. Hydrogen never has this 1 written next to it because it's hydrogen, and it would have 1 proton always. If it had a different number, it wouldn't be hydrogen. And that's really all we say about protons. They're positively charged, so hydrogen's got 1, helium's got 2, carbon's got 6, like that, and they're not changing. They're significant because they have that positive charge, so they are attracting negative charge, like that, and that's it. There's some really famous elements that have different numbers of protons and have, as a result, some different charge characteristics. So here's sodium chloride, sodium and chlorine, and in salt, when you dissolve it, it always dissolves into ions. We'll talk more about this, and the book talks about this. And what's going on here is that chlorine has 17 protons, it has a big pull, and when these dissolve in water, chlorine actually takes one of sodium's negatively charged electrons away because the nucleus of chlorine has such a strong positive charge. Again, we'll talk about that in some detail, and the book does too. So, bottom line, atomic number, boom, there it is. And it just defines the number of protons, which is specific to each element. The end. Okay, the next one we can look at is the mass number. And look, this is going to be telling us about the neutrons. And the way it tells us about the neutrons is it's the total number of protons and neutrons. So in the nucleus, are the protons and neutrons. And those have a mass. And the mass is about equal. So we can just add up the number and get an approximation to the mass, because it turns out electrons have almost no mass. So who contributes to the mass are protons and neutrons. They contribute equally. And so we can just do like this. We can say, oh, here's oxygen. It's got 8 protons. It's got an atomic mass of 16. So it also must have 8 neutrons. Like that, okay? So see, cool. We're dealing with protons, we're dealing with neutrons. And then here's the next thing, is that the atomic mass can change. So the atomic mass is often written here. And when it changes because the element is an isotope, which is a term used to designate radioactivity. And radioactivity comes from isotopes that are often unstable and will release energy and change the number of neutrons decrease the number of neutrons in the nucleus. So radioactive isotopes are active and degrading, and so we can see sometimes, like here's this P31. This one is common. There's another one that's common in nature, P32, and P32 is very radioactive because it's going to lose a neutron in the form of radioactivity and turn into P31. And in doing so, release this tremendous amount of radioactivity. Boom, like that. So our book talks about these neutrons because when biologists talk about neutrons, they talk about them in experiments. So in your natural life, walking around, talking, there's really no, neutrons are just not significant. The radioactivity is just not significant. Biologists use radioactivity in experiments, and they can be used to like tag a molecule. So it could be a radioactive molecule, and you could tag it, and you could monitor it as the molecule goes through your body. Monitor where the radioactivity is, and that's been important historically in biology. And another experiment is to do radioactive dating, carbon dating, and our book talks about that some. So the book talks about neutrons and radioactivity. You have to learn some more than I'm going to talk about them, but the book does a good job introducing a little bit of that. And it's cool because, boom, we're already done talking about neutrons and protons. Now, the next thing is electrons. And electrons often change and often do things because they're orbiting the nucleus. So electrons can be shared between two elements, kind of like if Mars came close to our planet, our moon would orbit around both. And that's called this orbital theory, where often in our book and often in biology, elements are described as if they're planets. And that's not perfectly accurate, but for biologists, it's a really good explanation of how atoms behave, like that. Okay, so we've got these electrons out orbiting the nucleus, and they are something that biologists talk a lot about. And it's not shown here, but this is the place where you'll see, I already did it once, you'll see, for example, that's going to show something like this. And so that's the position where biologists and chemists pay attention and keep track of electrons. And what we're saying here with a plus charge is we're saying that this sodium atom has a plus charge. Okay? What's going on is it has a charge of +1 because it has 11 positive protons and it must have 10 electrons. It lost 1 electron to chlorine when I used that example of sodium chloride getting dissolved in salt water. So that's how biologists and chemists keep track of the number of electrons. You can infer the number of electrons based on the plus charge. Magnesium, as an ion, has a +2 charge. How many electrons would a magnesium ion have? Well, it's got 12 protons, and it must have 10 electrons in the +2 state. Uncharged, the positive electrons have to be, positive protons have to match the number of negative electrons. So in this table, these are all uncharged, and by implication, this one has 3 protons and 3 electrons. Okay? Then over here, I said, oh yeah, when this sodium chloride dissolves, the chlorine is got a -1 charge. So 17 protons, it must have 18 electrons. Got 1 extra electron from sodium. And it's a good example to show how these can be affected and the electrons can be dynamic. Salt water, ocean is different than freshwater lake, and different because it has some of these ions dissolved in it that really affect the water chemistry, and we'll be talking a lot about water. So we're already getting just about done talking about the basics that our book talks about, introducing protons, electrons, and neutrons. Not more to say about electrons. Then, here's something. On our handouts, I made a handout, and it's several pages like this, and I'll use these in the PowerPoint, but these are just in a handout that I already handed out that are on our website for you to look through, because there's some just like terms to define, and not really to memorize the definition, but just to have working knowledge, because we want to talk about these all the time. So see on that handout, I'm talking about isotopes, I'm talking about ionic charge, so terms and concepts. Okay, like that. And so let me just scroll back here because, yeah, we're good. And so the next thing is that we've got the book, and then I'm talking, but I'm talking about stuff that's largely from the book, but trying to talk about it in a little different way. And then we got this chemistry handout that's on your website for you to go through as well, like that. And that's that. Okay, so the next thing to mention is that these electrons are, let's pick, well, let's go back to the periodic table and take a look at this. So these guys are uncharged. So this one would have 1 electron, 2 electrons, 3, 4, 5, 6, 7, and so forth. And see, there's an organization to the periodic table. It's got some kind of funny pattern to it. And there are rows. And the rows in the periodic table are designating electron orbitals. And orbitals are spaces or patterns around the nucleus. So there's the neutrons and protons in the nucleus, and the electrons are orbiting in a certain pattern. And the electrons are attracted to the positive charge in the nucleus, and but they're also, when they get really close, they're also repelled. And so they're orbiting at a certain distance and in with a certain pattern. And as electrons fill around any nucleus, there's some spaces or orbitals they can fill, and they fill them in order. So hydrogen has 1 electron, and it fills this first orbital, or these are called shells. And then helium adds another, and now this first shell is full. Lithium has 3, so the first shell fills, and then there's an electron in an additional shell. And there are a couple of shells on this row, and then this row is full. So over here on the far right, these outer orbitals are full in this column. So we start a new row because there's a new outer orbital. These outer orbitals are not full. The outermost orbitals are partially full, and the outermost orbitals have what are called valence electrons. So sodium has, see, it's just got one valence electron. Chlorine has 7 valence electrons. And it turns out that these outermost electrons are less stable, and so sodium would be happy to give up its electron to chlorine. I said chlorine's got a big pull, but also sodium isn't too happy with its one outermost valence electron. Look what happens here too. When chlorine gets that electron, its shell is now full. Something to point out here in this column is these are called the inert gases because they're not very reactive. They don't interact with other atoms because their outer orbitals are full. These want to interact with others, and what they can do is they can share electrons. So in the case of hydrogen, let's just do this right now. So we got H2O is water, and hydrogen would like to share its electron with something else and share electrons to fill up this outermost shell. Here's oxygen. Oxygen would like to share its electrons and share them with somebody who could provide 2 to fill up oxygen's outermost shell. So in H2O is pretty stable because in sharing their electrons, and the way that works is like I described, where our moon orbits around both Earth and Mars, because Mars is close enough. So these elements get to be a very specific distance apart because they're attracted yet repelled from each other, and then these shared pairs of electrons orbit like that. And that's often designated by a dash line, which is indicating what's called a covalent bond. In order to fill up their outermost shell, these guys would love to form a covalent bond with other atoms. And remember, we're especially focused on these guys, and especially these 4. And there's a rule. Hydrogen would like to form one covalent bond. Covalent bond is the sharing of a pair of electrons. Hydrogen would share one of its electrons with, say, oxygen. And oxygen would love to share 2 electrons, so it shares these pairs with hydrogen, 2 hydrogens. And so a covalent bond is the sharing of a pair of electrons, and these guys have patterns of bonds. So hydrogen would like to form one covalent bond to fill up its outer shell. Carbon would like to form 4, see, to move it over to there. Nitrogen, 3, oxygen, 2. When we get down to some of these lower ones, stuff gets weird, and that's where lots of chemistry is done. You know, we get down here and they're metals, you know, like gold and lead, and they have some different chemical characteristics. Like, for example, phosphorus would kind of like to maybe form 5 bonds. You know, so some of these rules start to be kind of bizarre, and it's because they start having such a big nucleus with so much charge that there are some different characteristics. And so phosphorus would maybe form 5 bonds and maybe have a negative charge as a result. That is the shifting between all of the atoms in that. So, you know, we're going to, in introductory biology, we're going to focus up here. You know, we're going to use these guys as little Lego blocks, and they've got these rules. And you'll see they form these covalent bonds like this. You probably have heard of methane gas, and there's methane. Boom. You probably have heard of methanol, and here's methanol. See that? Boom, like that. And carbon wants to form 4, oxygen 2, hydrogen 1. You probably have heard of ethanol. Whoops. Ethanol is 2 carbons. Like that. And so we're building small molecules already. And a molecule, as defined in our course, is atoms held together by covalent bonds. And that's in that handout. The handout just cranks through these, bottom line facts. It's cool because I have a PhD in biochemistry, and it's just a lot more of this. Just a lot. You can make like, make a model of a cell with Legos, right? Go for it. And so there's chapter 2 still, just starting to show that and showing that biologists and chemists draw this in some different ways. It's all showing the same thing, hydrogen held together by a covalent bond. And here's your definition, like that. Good. Here's water, and remember chapter 3 is going to be about water, and water is one of our big themes for our course. All cells exist in a water environment. So we're going to take a look at water, and water is special. That's a special molecule, and we'll find out why. Here I talked about methane. Here's a variety of simple molecules that fit those rules. That's molecular oxygen, like the air you breathe, is not just a single oxygen atom, but oxygen's held together by this is called a double bond. So that would be 2 pairs or 4 electrons are being shared. Why? Because oxygen would really like to form 2, like that. These have physical space. These occupy physical space, and that's why that Lego story is a good one, because Legos fit together in a certain way. You can see these things starting to fit together in a certain way, and we can build bigger molecules, and they're based on the rules of these little molecules. Here's some rules. That distance is constant. The distance between atoms in a covalent bond is constant. The covalent bond does not bend. So it's like a little toy, a little stiff toy. Significantly, it can rotate. These atoms can rotate. So see how this is like a 3-dimensional atom structure here? So imagine if that was the case up here. So we have just a little bit bigger molecule. And now rotating around this pivot, see, this guy would be moving relative to those. And so when we build a protein like insulin that's thousands of atoms together, there's quite a bit of flexibility. Where does the flexibility come from? Exclusively from that rotation. Because each of these can rotate. So you start chaining together stuff, and at first it becomes kind of a stiff thing, and then it becomes just so big that it's just like cooked spaghetti. Like that. It's very floppy. It's held together, chained together, because covalent bonds are really stable, but it also has some bending, not bending, like twisting characteristics. Just showing this again from our book and then from our handout. The next thing is that we've got these 2 types of covalent bonds. And what's going on here is that we already talked about how sodium can lose an electron to chlorine. And it does that because of this filling up the shell and emptying the shell. But I said the very first thing is that, and I think these numbers are right, the main reason is because the chlorine has more positive charge, and it pulls it, and that quality is called electronegativity. And electronegativity is defined up here. So chlorine is more electronegative, and it will pull that away. Okay, so now I'll go back to here and just show you this on the periodic table, that as you go down the periodic table and over on the periodic table, electronegativity changes. As you go over in this row, you're increasing the number of protons, see them? And so you have an increased electronegativity. Oxygen is more electronegative than carbon because it has more protons in the nucleus. Okay? Like that. Now, the next thing is interesting, a little bit surprising at first. When you go down the periodic table, you get decreased electronegativity in the valence shell. And why is that? because the valence shell gets further and further away from the nucleus. So there's less pull, right? So I said for any nucleus, you first fill these inner shells, and then you have to fill some outer shells. And the further you go down the table, right, each row is another set of orbitals further out. So as you go down the table, you're getting further out, and there's less pull. So it turns out that fluorine is the most, it's just nice to remember this, even though fluorine's not a very significant biological molecule, it's the most electronegative of them all. Why not these guys? We already said these guys are inert. We're talking about covalent bonds and the desire to interact, and these guys don't. Their orbitals are full. So we think of these as inert and having really essentially no electronegativity because there's no place where they want to put an electron. Fluorine's got, they would really like to have one more electron, and it's got a lot of charge, and it's got an empty spot in a orbital that's pretty close to the nucleus. Like that. See that? Okay, so oxygen is this, basically the second most electronegative element. So think of oxygen as having a strong pull. Okay, so now the next thing is we go back to here, and we get to define two more terms, and that is we got two types of covalent bonds in biology, and they are nonpolar when the atoms involved have similar electronegativity, and they are called polar when the atoms involved have different electronegativity. And significantly in water. Hi.
You know where
yeah.
Tracy's room is?
Yeah. I'll be right back. It's in chemistry. Do you know where chemistry is?
Oh, is it like part of the building?
Yeah, it's over. It's on the third floor. But
Okay.
you have to go down and back up. Yeah.
So down, like another side.
Yeah. So that's the easiest thing is go over to the first floor. Go over to the first floor. But don't go that far to the north. Go to the second floor. It's more than you can that.
Okay.
Yep. All right. Yep. All right. Sorry, I'm back. I had to give some directions. But it gave you a chance to think. And think about what? Oxygen, when we go back to the periodic table, oxygen is more electronegative. So yeah, it's going to share electrons with hydrogen, but it's going to have more pull. And so the shared electrons are going to be hanging around oxygen more. As a result, there's a little bit of a partial negative charge around the oxygen, a little bit of a positive charge. And that's why it's called polar, like poles on a magnet, where you have a positive and a negative, kind of like that, a little bit of feeling like that. As a result, we're going to learn about in a second, this is what's special about water in Chapter 3, is that in a cup of water, the water molecules are attracted to each other. That's intermolecular. And that's one thing that makes water very, very special. It's just that, boom, we've learned so much biological chemistry already. And I think there it is, so showing in our book and still in Chapter 2, this idea of a polar covalent bond. And we're going to stop here, and the next mini lecture is going to be about water. And because we use that to build that water environment, but also to start building molecules with polar and non-polar covalent bonds.